Thanks everyone for attending this tinyML talk. Today we have a great pleasure to have Paolo Bombelli from the University of Cambridge here, who is going to talk about an algae-based battery, which is capable to power low-power microcontrollers. The title of this presentation is Bio Photo Voltaics, BPV. Is a term that we're going to hear quite a lot during this presentation. And it's going to talk about the fundamental principles, and it's going to show us practical applications of this fascinating technology. Here we are in the Cambridge office of ARM. So hello everyone, I'm sure if you can see us, but we have excited to have here Paolo, and yeah. So let's start. Let's start with the housekeeping. And first of all, I'd like to thank you all our strategic partners for behind the tinyML. So here I'm not going to list all of them, but it is definitely we need to give a big thank you to them for making this possible. So one thing, definitely, I'm looking forward to this event. I'm gonna be there in person. Is the tinyML Innovation Forum 2022. So the event will be held very soon in October 10th to October 12th. The keynote speakers definitely excited to see the presentation from Massimo Banzi, CTO of Arduino, and Alberto San Giovanni Vincitelli from the UC Berkeley Cadence and Synopsys. So definitely I'm looking forward to see you there in person and to have to learn more from this event, in-person event. Okay, so if you're new in this forum, you haven't attended any meetups groups, so I recommend to register to our meetup group to be always up to date to presentations, events that we are organizing in the UK regions, but at the same time, you can register to the global meetup group where you can learn about the other events that the tinyML Foundation is preparing, organizing worldwide. Don't worry if you're new to this field, because there is plenty of material where you can learn from scratch what this technology is and what it can bring to human society as well. Here is the link where you also can subscribe to the tinyML YouTube channel for where we host all our events, recorded events, including the one we are going to host today with Paolo. This is definitely the resource where you should look at to learn more about tinyML, whether you are a new buyer or an experienced developer in the tinyML space. And now it's time. So I'm very glad to introduce Paolo Bombelli. I'm gonna read his bio. Paolo holds a teaching position in the Faculty of Built Environments at the Bartlett UCL in London, and is a visiting researcher in the Department of Biochemistry at the University of Cambridge. He obtained his PhD from the University of Cambridge in 2012 for work on the fundamental principle of bio-electrochemistry. Since then, he has gathered around him a team of collaborators from several leading research groups, industrial partners, and non-profit organizations. By combining biology, material science, and engineering, his long-term goal is to translate the fundamental principles of photobioelectrochemistry into practical applications. Please welcome Paolo to this event. And Paolo, 8%, yeah.
So first of all, thank you very much, Gianmarco, Dominique, and Olga for organizing it. Thank you, everyone, to be here. So as Gianmarco said, during my talk, I will use quite a bit this acronym, Bio Photo Voltaic, BPV. So therefore, probably the best way to start is to give you a reasonably simple way to explain this this acronym. So a biophotovoltaic is a hybrid system combining together a biological photosynthetic component, like an algae, with an electrochemical setup. So before telling you more about how this system works, I decided to give you a quick overview of what we can do with those hybrid systems. So first of all, we can use this hybrid system as a tool of investigation, given that the electrons generated by the biological components are taken and measured by the electrochemical part. The way that this current emerge is telling us something about the metabolism of the biological component. Therefore, we can actually use the electrical signal for probing the metabolic activity. This means actually having a very powerful tool of investigation. The second possible application is actually using this hybrid system as a biosensor. And I want to stress that in this specific example, we are not going to use them to power sensor, but the system themselves is the sensor, in the sense that if we are having the system receiving some toxic component, like this example is an herbicide, atrazine. Atrazine might compromise the metabolic activity of the photosynthetic organism, means compromising also the electrons generated and therefore the current output. Therefore, by analyzing the profile of current output, we can actually detect the presence of the toxic in the environment. A third example is actually linked with the possibility to change the metabolism, also the gene expression through electrical signal. Again, as we create a hybrid system, means that the biological components is talking with the electrochemical part via current, but also means that the electrical part can play a role on the life of the biological components using electrical signal in the form of bias potential or bias current. Therefore, we can actually trigger gene expression using bias potential and bias current. And it's exactly what we have done in the recent study, where we were able to turn on a gene responsible for the production of fluorescence that we were using as a marker. And finally, we can generate electricity. We can use this system to generate electricity. So during this talk, I will focus on this application. Generally, two questions are coming to me when I present biophotovoltaic. The first one is how does it work, and the second one is how much energy does it generate. In the next few slides, I will try to answer those two questions. So the first one, how does it work? So first of all, we need to have the two main components, which are the cyanobacteria or algae, which are the photosynthetic component of the system. And the second part is the electrochemical part, which is in a very simple way made by two electrodes, which are arbitrarily called anode and cathode, connected together by an external circuit. The photosynthetic organism is in charge to conduct all the processes that collectively are called photosynthesis. Means harnessing light and using light to split water in oxygen, proton, and electrons. This is generally called water photolysis. Oxygen is released in the atmosphere, and proton electrons are then combined with carbon dioxide through a quite complex set of reaction called dark reaction or Kelvin cycle to transform carbon dioxide in organic carbon. Starting from very simple sugar, then to pass through carbohydrate, for then build a lipid protein and whatever you need for the life of the organism. Then some of those metabolites can be respired, so can be reoxidized, means regenerating the electrons and protons that they were originally consumed for building up the metabolite. And some of those electrons can actually be excreted from the photosynthetic organism and injected into the anode. The process of moving electrons from the intracellular space to the anode is probably one of the most challenging elements, part of this system. And I will get back to this point in a couple of slides. At the same time, protons, or probably it's better to say protonated ions, protonated water, can diffuse from the photosynthetic organism in the space where those organisms are located. So having electrons injected into the anode doesn't really explain why those electrons should move. And the reason to move is based on the thermodynamic property of the anodic and the cathodic reaction. So having a cathodic reaction that is more positive compared to the anodic direction creates a gradient and explain why electrons are moving from the anode to the cathode. To make the system a bit more directional, we can also introduce a barrier, a proton permanent barrier in front of the cathodic element of the system. So in this way, we permit the protonated water to move to the cathode, but we stop other elements to mess up with the cathodic surface. And finally, in order to make the system more rapid, so because thermodynamics explain why the system works in a certain direction, but it doesn't tell us too much about the timing of the system. So in order to make the system more rapid, we can provide a catalyst that make the cathodic reaction more favorable, more in terms of kinetic property. So these are the basic components in a very simple way that constitute one of these bio photovoltaic systems. As promised, I want to go back to tell you a bit more about the electron transport that permit electrons to move from the intracellular space to the anode. I said to you, this step is one of the most challenging steps. And there is throughout the last 10, probably 15 years, several ways to permit these electron transport have been discovered and characterized. We can divide them in 3/4 groups. The first one is via a soluble electron mediator. So something that is soluble in water and is able to exist in oxidized or reduced form. It is therefore able to pick up electrons from one side and deliver to the other side. Example of those are the ferrisanide or phenazine, for example. Another possibility is via protein located on the external membrane. And some of those proteins can actually live, they are called transmembrane protein in a sense that they are located across the membrane. They form a sort of a bridge from the intracellular to the extracellular space, picking up electrons from the intracellular space and delivering those electrons on the extracellular space. A couple of examples are the ferroreductase or the NADPH oxidase. There are other elements are being characterized mostly on non-photosynthetic organisms, which are generally called conductive pili or conductive filaments, are nanomicrostructure protruding from the cell, and they are proven to be electric conductive in the sense that they hold redox couple, and electron can hop from one to the other, moving across and literally acting as nanowires. Finally, we can also have a sort of a mix of those three mechanisms. Plus, we can also consider that cell frequently produce something called extracellular matrix, quite complex mix of metabolites, sometimes sugars. They form a sort of a glue, gluing together cell. Within this biological glue, we can have a redox process, which actually make the process of electron transport more favorable. So I want to give you an actual example of what I was telling you just now. So on the top left, you can see those filaments are anodic material on which photosynthetic organisms are being left to form a biofilm. So the photosynthetic organisms are colonizing these filaments made by metal. In fact, on the right, you can see a magnification of one of these filaments, also surrounded by this green jelly material. We took some sample and we characterized this, and we found a very complex mix of photosynthetic and non-photosynthetic organisms. You can see on the right of this slide the list of the microorganisms that have been identified. So some of them, like the bottom one, this one called Snecocystis, is a photosynthetic organism that we initially introduced. Other, they have been coming in via environmental contamination, given that we were leaving this system running under non-sterile conditions. So we were permitting contamination. And we have done that because some of these organisms are actually known to be able to be electrogenic, so be able to exchange electrons with an electron, like halomonas, for example. Therefore, we propose a mechanism of electron transport. You can see on the cartoon on the bottom right of these slides, where either the photosynthetic organisms are able to inject electrons in the electrode, or the photosynthetic organisms are feeding metabolites in the non-photosynthetic one, which are in charge to do the process of electron transport. So by building up a system using these characteristics, we were able to actually powering a microprocessor, precisely in our core Cortex M0, and running it for 6 months. This is actually what we were able to publish. In reality, the system ran for longer than that, ran for over a year. So the system was very stable, was fed only with natural ambient light, and was run under ambient condition, means we didn't control the temperature, we didn't control, so we let the system working in a way that is mimicking a proper application. So the second question that frequently comes to me is how much those systems can generate. So I'm giving you these two examples that we published no longer ago. The first one is based on a biophotovoltaic built into a microflow based design. So it's a tiny system, and with this one we were able to reach about 50 microwatt per centimeter square. The second one is the one that we discussed before, and this one is a more macro scale device. And with this one, the power output, the peak power output is about 4.5 microwatt per square meter. Therefore, either way, we are in the region of microwatt per square meter, which is not necessarily very much. Therefore, we go to the point is what we can do with this microwatt of power. And to answer this question, I try to make a sort of a consideration that electrons are all equal, but electrical energy is not. In a sense that, so if we think about in terms of how much we pay for electricity, I know that is a topic now, given that the price is rising. So I try to be quite realistic based on very recent price, at least here in the UK. You can pay for kilowatt hour electrical energy from the national grid between 0.3 and 0.5 pounds per kilowatt hour. On the other hand, if we generate electricity from batteries, the price is 1,000, even 10,000 more expensive. Therefore, microwatt can be valuable depending what you power and where this powering is used. For this reason, we start mapping a possible domain where the biophotovoltaic might play an important role. So we define three domains. The first domain include devices which require less than 1 milliwatt to operate. Typically, all the microelectronics and to be honest, in this domain, if we look, I'm pretty sure with some of you will have example, even something that you work every day. The second domain include the devices that operate with, say, within 1 watt up to 1 kilowatt. So for these devices, we think that this might be the domain of biophotovoltaics, perhaps not today, but in the near future. So we can reach those power output by improving the technology, we can improve the biology, but also we can improve the physical elements of the system. And finally, I think there are elements that in our view they'll be outside the domain of biophotovoltaics. Typically large domestic appliances, so industrial electric devices. For those, I don't think the biophotovoltaics will provide a meaningful source of power. Thinking about so the so-called microelectronics, we need to face the reality that there are other technologies that can compete with the Bio Photo Voltaics. And I list three of them at least. So batteries, classical photovoltaic, and fuel cell. So in terms of current density, I think batteries definitely, they provide more current density. Photovoltaic depends. I think I said about 10 milliamps per centimeter square. I think is a quite generous, especially for the commercial one. And also bear in mind that generally that value can drop quite a bit if there is no light, down virtually to zero. Fuel cell, they generally have a good current density. For the biophotovoltaics, as we said before, the current density is still limited below 1 milliamps, depending on the size. But if we associate this with a capacitor, we can have short bursts of more intense current density. So in terms of the energy demand, energy on demand, battery, definitely you can have the energy when you need it. For the photovoltaic, unless they are combined with batteries, energy is linked with the presence of light. For fuel cell, indeed, you can have it when you need it. And again, for the biophotovoltaic, this is one of the advantages. Because if you don't really require electrical energy right now, you can actually let the system resting. The biological components, they can build up metabolites in the cellular body and then giving you this energy back when you need it. So there is a sort of internal biocapacity. In terms of to be renewable, I know there are people working on more sustainable batteries. I know, and I'm pretty sure good results will come out, but not necessarily all the batteries are considered environmentally friendly. This consideration is about having a renewable source of energy. Clearly, a battery will run out of power. Of course, you can recharge it, but you need a source of power to recharge it. Whereas photovoltaic are virtually endless, given that keep converting solar power. Fuel cell, they need fuel, but this fuel can be waste product. And for biophotovoltaics, as we said before, the source of energy, the primary source of energy is the light. And finally, environmental consideration for batteries, as I said, it can be plus and minus. So some batteries are more environmental friendly than others. Equally for solar panel, especially the production of solar panel not always is environmental friendly. Whereas for fuel cell and biophotovoltaics, pretty much all the components are environmental sustainable. So I want to finish just to summarizing what I told you today. So combining biological photosynthetic system with electrochemistry, we can create hybrid devices. And those hybrid devices can be used in different domains. For scientific investigation, to be used as an environmental sensor, to manipulate cellular metabolism and gene, and to generate electrical current. When we generate electrical current, the current performance remains below 50 microwatt per centimeter square. So at present time, we can power small electronic devices, for example, for IoT application. But we can actually think that in the future, performance will go up and we start generating power output in the region of milliwatts. And this could permit to range more substantial electronic, like to charge mobile phone, small light, also power laptops. I want to say thanks to Chris Howe, all the people in House Lab. Emery and Anand that from ARM, actually, they helped very much with the project where we power the microprocessors. All of our sponsor, the generally support the project. And thank you for attention.
Thanks a lot, Paolo, for this fantastic presentation. I'm pretty sure we're gonna have quite a lot of questions, but before the question, if you can see these, if you can see us, here there is actually an algae battery-based battery, right? You wanna talk about?
Oh yeah, okay. So I brought this as an example. It is fully functional. So these the active components are inside of this internal body.
So, bigger, right? Perfect. Yeah, I think. Oh, nice. Very good idea.
So the body is made by acrylic. It's made see-through in order to actually the light to penetrate. The algal material, specifically those is based on cyanobacteria, are based on this area. So, and this actually act as an electric connector. So the anode with the bacteria are inside of this chamber. So there are two ports that in this moment are covered with tape to avoid it to drip out. So on this side, there is the cathodic end of the system. So if you remember, the cathode to operate required oxygen. This is why the cathode, which is this black layer, is actually exposed to atmospheric oxygen. As I said, it's fully operational.
Let's do it. I mean, we can try at least. I don't know. We need to get
one
closer.
person holds the battery.
Yeah, we need to get closer to the camera. Okay, so.
So this is measuring a
bit up, yeah.
Potential, and if we are lucky.
Yeah, 840, something like that, 850.
Yeah,
48.
that is a millivolts. It's pretty much what you can expect, given the thermodynamic property of the cathode. Yeah. So yeah.
Thanks a lot. Thanks a lot. I mean, as I said to Paolo when he arrived, well, actually, I didn't expect to see something that small, because for me it is small, and it's very cool. So let me see the question. Before the question, actually, let me share my screen again. First question is, does the anode cut material suffer corrosion or oxidation?
Yeah, it does. So this is why we need to choose the anodic material carefully. So there are there are material. We've been testing several materials as anodic component, and actually is one of the key elements that need to be chosen in order to interact with the biology. For example, we've been testing carbon fiber, which is quite resilient to oxidation. However, for some reason, it doesn't really work well with forming a stable interaction with biological and with photosynthetic organisms. On top of this, it tend to be dark. That means on the optical level, it doesn't really give you any advantage. We test some oxide, like indium tin oxide, works well on the lab level to doing experimental work. But it's not proven to be a great material for application. In this specific example, we've been working with aluminium. So aluminium is reasonably stable in alkaline condition. And when the photosynthetic organisms start consuming carbon dioxide, they deplete the environment and they create an alkaline environment around the pH, between pH 9 and pH 10. These trigger the formation of aluminium oxide and hydroxide on the surface. In fact, on the initial part, part of the operation, we can have some we call abiotic current derived from the oxidation of the system. But then these abiotic currents start dying out, and we start having electrons from the metabolic activity. So indeed, the short answer is we need to be very, very careful what we pick as anode.
Okay, thanks, Paolo. The second question is, does it work better in indoor bright light conditions? And if so, how much better?
So, we need to treat those systems as a plant in the same way that we have a plant at home. So the plants require light. So if you keep your plants in a box for too long, will eventually die due to starvation, lack of light. So in the same way, you need to make sure that those plants they receive light. On the other hand, too much light could actually cause some, it's called photo stress. So not necessarily a lot of light will help the system. The indoor versus outdoor, we've been testing the operation of the system under different temperature. Indeed, if you go too high, you might kill the organism. So for too high, I mean, if you expose the system for temperature above 50 degrees for a long time, at least with this type of organism, they will die. You might get better result if you colonize the system with organisms that are already used to high temperature. There are some bacteria that live on the geothermal vent. For those, definitely you can operate at high temperature, but then they might not operate well at low temperature. So there is definitely an optimal range of temperature depending on the organism.
Okay, I have another question, actually a follow-up question. What about low temperatures?
So again, we test actually sub-zero temperature, and so the formation of crystal of ice will cause the system to stop working, in a sense that there is no current anymore coming out. But then defrosting the system, the operation will recover. It will take some time, but it will recover.
So thanks for the question. So there is another one here. How does, probably you have already answered, but I'm gonna read the question anyway. How does temperature affect the output power? Can high light flux overheat the biological component?
Just repeating a very good way, absolutely yes. So depending on the biological component, there is a sort of an optimal.
Okay, thanks. Any questions from Cambridge office? Yeah.
So when algae dies or eventually runs out of power, is it easy to kind of replace it? Do you have to replace entirely the whole system, or you can just replant some?
Okay. So ideally, so when you say when they run out,
yeah.
In principle, the fuel is light.
Yeah.
So if you keep providing light, and you can assume that the population will stay somehow stable, in the sense that in this device, which the active chamber is about 1 centimeter square, there are probably few billions of algae. So at any time, few thousand probably are dying and others are replacing those. So if the overall balance create a stable community, you can expect this stable community will live for quite a long time. In fact, we do have a stable community that have been living for more than 3 years. Now, telling you what's the limit of this, I don't really have any experimental data to tell you. At least for 3 years, we'll be able to prove that they are able to operate in a stable way. And end of life, this system can be completely recycled. So you can open, disassemble the different parts and recycle.
How long does it take to build this battery?
Okay.
In laboratory.
It's probably, I would say, so between 1 and 2 hours to build up a nice build system. On the other hand, so I've been using these devices to doing outreach activity in school, and generally I bring with me all the parts and build up devices with students in a couple of hours.
We all want to go back to school now.
Maybe I should have.
Next tinyML you came in.
And I need to say that generally 80% they actually work, which is pretty impressive given that, I'm doing this generally for students that are at least, say, 12, 13 years old or older, because otherwise it get a bit too difficult even to interact. And not necessarily all the components are understood by the students. But if they're following what the instruction, generally the result is fully operational.
I'm going to click this. So actually, it's kind of two combined questions. So in that first slide, you obviously said, or early on, you had the series of different use cases that you were looking at. And you said you focused on the battery one, which is fine. I hadn't realized there were other ones. So I guess the first question was, we talked about essentially turning an algae on, essentially using, in a sense, doing this the other way around, sending current into an algae. Now, I'm no hardware engineer, and I'm surrounded by them, so we have to be a bit careful here. But this is sounding quite a lot to me like a transistor. So is that a possibility that you could bioengineer effectively a biotransistor essentially rather than a silicon one?
Okay. My understanding, myself not a physicist, but my understanding is the core of the photosynthetic apparatus, so the works similar to a transistor. The entire photosynthetic system or the entire photosynthetic organism is, I don't know how much similarity you can find on the functional level. On the other hand, the fact that you can interact via electrical stimuli open to at least to investigate, to explore what you just said. So I'm not really sure. I don't really know, but at least there's a possibility that can be explored.
So the second half of the question was the other picture, which was where you talked about detecting herbicides, which I think is really, really relevant to tinyML because it's essentially, you can see it as a sensor that we can then learn something from, learn how it reacts to something, and then start to make decisions. I'm not sorry a question. It's just a really interesting observation. I think that's a really appropriate use case for tinyML. There are some more questions coming, Jaco.
Yeah. So I'm gonna take another question from Zoom. The other one is you mentioned power output of 50 microwatt per square centimeter. Can this be scaled up to a square meter to give approximate 0.5 watt, or is it scaling too difficult?
Specifically that one that was built on a microchannel system is well, it's scalable, but I don't think that we are planning to go through that process to scale up that system. We are more planning to scale up what I've shown you here and try to improve the performance of this and reaching that 0.5, perhaps even 1 watt square meter by using technology more like this one. So it's scalable, but I would not build on the micro channel.
Thanks, Paolo.
Sorry.
Yeah.
So what do you mean by micro channel?
So it was so one of our, we run some devices where actually we integrated all the electrodes in a micro channel. So I think it was about 6,000 micron long, 25 wide and 25 high, and we integrated inside the electrodes. We did it because in this way we were able to minimize the mass transport of the positive charges and using time as a proton barrier. So it was, in my view, a very interesting experimental academic exercise, but not necessarily sure how much this can be translated in a large scale. But I might be wrong because I know that sometimes scaling up micro devices is amazing.
Okay, taking other questions from Zoom. Could it be a growing community instead of a stable one?
Absolutely, yes, in the sense that so what we do is so we provide a starter more than a starter, but then we let the community evolve. In fact, we have collaborator based in Oban, in Scotland, and is a marine biologist, and what this person is studying biomes, so the combination of microorganisms. So definitely letting the community of evolving is one of the key elements to have a solid and long-lasting system.
Thanks. Okay, another question. Could one feed them with organic material?
I guess the answer is perhaps yes, but then you're transforming this system in a so-called microbial fuel cell, which is anyway great. I mean, something that I didn't mention is this system, some people could also call it a photosynthetic microbial fuel cell, which is equivalent to this, but operating only with bacteria fed with organics. I don't have anything against it, just at some point the organic will run out. So it's more convenient feeding light, which is
Okay. Would a completely non-human visible light source, for example, UV, something that could be tailored?
Okay, this is
what part of the light spectrum actually is more sensitive?
I mean, so given that this system operates with, say, a photosynthetic organism, they operate, they absorb light thanks to pigments. The most common one is chlorophyll. However, specifically for cyanobacteria, there are other pigments called phycobilosomes that will absorb more on, say, between 550, 650 nanometers. But for chlorophyll, there are the two main peaks in the blue and the red, with a gap in the green. So theoretically, the most photosynthetically active radiation are the red and the blue, with predominance on the red. If we are thinking to try to find a way to expand this, to use the UV, it's quite hard. Generally, they generate a bit of damage to the biology. If we think to use the infrared, except if you operate with specific microorganisms that are actually able to harness the red is not, the infrared doesn't carry enough energy. So I would say the visible light is the natural food for those systems.
Okay, thanks. So, any other questions?
What kind of theoretical limiters are on the power you can generate in like a single centimeter square cell or something?
Okay,
like that?
we've done some calculation, and I mean, what I'm giving you now is not the theoretical limit, is what we think might be achievable under very optimistic conditions. So we believe that we might be able to get around 5 watt square meter.
That's pretty tasty.
Yeah, exactly. That is, by optimizing all the elements. That is so what we believe that is possible. It's not the theoretical limit, but the theoretical limit sometimes I even don't like to discuss.
No, the theoretical limit is the amount of light hitting the surface.
That is actually, well, theoretically you can have, even in the UK, you can have like a 150 watt square meter of light coming in. But I think in a very much optimized system, you might be able to get 5 watts.
So my question is, do you think it's ready for real-world application? For example, can I connect a Cortex M to it and get sensor readings for 6 months, something like that?
Well, so it's actually what we've done we connect a microprocessor from ARM for a year and getting continuously. I think we were performing very simple calculation. If I'm not mistaken, we're just +1 + 1 = 2. Is it true? Do it again. It's not true. Tell me that something is wrong. And we were able to do it continuously for 1 year. So in principle, the answer is yeah. Now, in my view, the challenge is to find a meaningful application where we can actually develop a customized system and test it. And exactly where we are now.
Maybe a follow-up question for me is, you said about the next steps, right? We like to have a million-watt algae-based battery. So yeah, and definitely for me, this means that it's going to open, quite a lot of use case, tinyML applications or microcontrollers. What is currently missing for being there, and what we are exploring to reach that target?
It's probably, I would say that if I need to bet on what would really improve the performance is be able to characterize, improve the process of electron transport between the photosynthetic organism or the organism and the anode. Given that the machinery to harness light and generate electrons has been evolved for about 2 billion years, is actually quite efficient. So the ability to generate electrons is there. What is not there is the ability to transport those electrons from the cellular body to the electrode, because for nature that step is not important. So if we are able to improve, and we've been doing a lot of work on that domain, but if we can keep working in that domain, we might be able to reach that target.
So we need a breakthrough in material science.
Again?
Do we need a breakthrough in material science?
We need to carry on because we've been doing some breakthrough in this domain. So using the current material is already a big improvement compared like 5 or 10 years ago. We need to keep working on that domain, yeah. And somehow be able also to combine microorganisms that are photosynthetic with microorganisms that are not photosynthetic, but they do possess intrinsically the ability to transport electrons. This might actually create a very productive mix.
Question. What does your system that you mentioned with your microcontroller currently look like? Is it just a development board you're powering with this battery, or is it a chip that you have?
So can we, let's try. Yeah, you can.
So we use that system that you see on the top left of the slide. So it's just marginally bigger than this. Well, the geometry is a bit different, but
it's basically just this bolt battery wired to this development board.
Correct. Yeah. So bear in mind, we were not powering the entire board. We were powering only the processor. The board was powered externally.
So power?
Externally. So I mean, we had like an external source of power for the board, and we were powering only the processor.
Why? Why external power for the board?
Because unfortunately, to power the entire board was not.
I suppose the board has probably display and other things, other peripherals. Microwatt is not enough, but definitely microwatt. We are in the range where on the CPU in sleep mode, for example. A CPU for a microcontroller in sleep mode is on a microwatt range. So, and definitely this kind of battery can keep it on unless there is an interrupt, and probably we need another battery to supply the correct power. There was a question, I believe, in the QA. Yeah,
Viet has one question.
Yeah, I have one question. So, what happens when the battery is overloaded? Like, when you have a low, very low, does the algae die?
I don't really have an answer in the sense that we have not tried to stress the system under this situation. I don't really have a good answer. I think it's something that probably we should try to explore.
Okay. The question we have here: how do you monitor the health of the cells in the device?
So there are specifically, given that we're talking about photosynthetic microorganisms, we tend to use a parameter that is called autofluorescence. That is related is a fluorescence signal that is related with the activity of the photosystem too. So I mean, when you have autofluorescence, means the photosystem 2 is operating. And thanks to this parameter, you can actually assess the efficiency of the photosystem 2 that is proportional to the health of the biological component. So this, or alternatively, you can measure the rate of oxygen evolution, which is a bit more complicated things.
Thanks. Any other questions?
So you were saying these could be built by school kids. Does that mean that there's no? Does that mean that it's actually relatively simple and there aren't any like highly toxic chemicals in there that would? And that's the main advantage over solar power.
Is one of the main advantages. So yeah, and also the fact that you can build it, and if something goes wrong, you can fix it. So where the classical solar panel, there's nothing to do if something goes wrong.
You throw it away if it goes wrong.
Okay. 3 months we're gonna have another tinyML session.
Yeah,
Like
we bring over some. We're gonna actually build together. Yeah.
So first,
oh no, I'm just going to ask you a question about cost, but I think it's related. So I think it's rather low cost just to build a system like this.
I mean, it's relatively low cost. So this probably will have cost me, let's say, a few pounds. So and probably just because I pay a lot for the plastic.
Yeah. You can just give it to school kids to build them.
Well, we normally bring all these to school kids.
any other questions? So if we don't have any questions, I'd like to thank again Paolo for his brilliant presentation. The remaining slides. First of all, I'd like to thank the tinyML Foundation for making this possible, and the tinyML UK group for the support for giving this presentation today. As I said before, we'd like to thank again our tinyML strategic partners. In this slide, we have reported all our partners that are behind the tinyML. And so here there are the slides about the execute strategic partners. One Arnold, who today also hosted this event. And also, just a reminder, in case you are interested in AI talks, there is the AI Virtual Tech Talks. You can register to learn more about it. Edge Impulse, the leading development platform for Edge ML. Qualcomm, with Qualcomm and AI research for advancing AI research to make efficient AI ubiquitous. Xintian, making Edge AI a reality. In terms of Platinum strategic partners, we have DeepLight, with the fastest video analytics solution on ARM CPUs. Key Leica Tech, global IoT solutions. Reality AI, tinyML software that covers the full engineering lifecycle, Reality AI tools. Renesas is enabling the next generation of AI-powered solution that will revolutionize every industry sector. Sony Semiconductor Solutions Corporation. And also we have the gold strategic partners, Analog Devices, PhotoHub, making over-the-air firmware and ML models updates simple and accessible. Microsoft, NXP, Seed Studio to deploy tinyML into real world. SensiML with analytics toolkit suite. ST Microelectronics to provide extensive solutions to make tinyML machine learning easy. Synaptics, engineering exceptional experiences. SynSense. And here we have also the silver strategic partners. So AZIP, our own devices, EMSA, Green Waves, Gravity Inc., EBM, ImageMob, Item, RS, NotAI, OctoML, Prophecy, Kixo, Rixin, SAP, Silicon Labs, Stream Analyze, TDK. And that's it for today. So on behalf of the tinyML UK group, I'd like to thank you for attending this presentation. Thank again Paolo for his presentation. Hope to see you again to our next tinyML session. Paolo, we are ready for building algae battery base. So thanks a lot and have a nice evening or have a good day. Bye-bye.